Hello. Today we're going to be looking at the carbohydrate fermentation test. Last time we did the extracellular enzymes. So in the series of labs that we're going to do next, we're going to do the intracellular enzymes or the endo enzymes. These are the ones that are inside the bacteria. They are not excreted from the cell. When the sugar comes into the cell, usually broken down by an exoenzyme, then the sugar can then be broken down further for cellular respiration or fermentation. So what we're looking specifically at today is fermentation. And remember, we're talking about these biochemical tests because we're looking at the identification of the microbes. These are looking at the particular enzymes that they have. And recall that fermentation can happen in the presence of oxygen or without oxygen. Oxygen is not actually the factor that causes a bacteria to always go into fermentation. Some microbes will naturally ferment, others will not. Some will ferment after aerobic respiration, so it just depends on the microbe. So when we talk about fermentation, recall that fermentation uses organic molecules to reduce the hydrogen ions and the electrons that are at the end of glycolysis. And also recall that it doesn't go into the Krebs cycle and it doesn't go into the electron transport chain. And with fermentation, remember that the end products of fermentation are either going to be an alcohol, an acid, and possibly gas. Well, in the test that we're doing today, we're looking specifically at the presence of acid and the presence of gas. Now with the fermentation tests, it's really easy to detect fermentation because we can use an indicator called phenol red. And phenol red will turn yellow in the presence of acid. And it will also turn more of a pink color in the presence of a base. So in today's lab, when we do the test and we look at the results, what you're going to be looking for is a color change. So the original tubes are going to be a light red in color. If they're not red or very close to red to begin with, then there's possibly a contamination or a problem with the media. So make sure you note the color of the tube before you even start. The other thing about these fermentation tubes is they have inside of them a small tube. Now you can't really see it, but if I shake it, you could probably hear it, okay? In the tube itself, upside down in the tube is called a Durham tube. And the Durham tube also has the same liquid inside of it. And what the Durham tube will do is if the bacteria produces gas, like CO2 or hydrogen gas or something, then the gas will get trapped in that Durham tube and you're actually going to see a bubble inside of it. So for these tubes, because they have the phenol red, which is the pH indicator, and the Durham tube, the tubes can actually detect both the acid and the gas production. A bacteria may produce acid but not produce gas, or it might produce both, or if it doesn't ferment that sugar, the tube will remain the same color. Now, one of the things to keep in mind about all of these tests, when you look at the results, you actually want to look for growth before you ever determine results. So if after the 24 or 48 hours that the tube has sat in the incubator, if it's still red, the first thing you want to look for is turbidity. Look for is there a pellicle, is there sediment, is it cloudy, okay? You're going to have controls, so you'll be able to compare to the control to see if there is a change in turbidity and also to see what the color change is. The other thing is I mentioned that it has a basic reaction also. So if it actually raises the pH because it doesn't use sugar, but it might use the peptone that is in the media, it might turn more of a pink color. That's not a positive reaction because we're looking for fermentation, which will go acid, okay? So there's a lot of different results. This is a very differential type of media and it could tell a lot of different things. So for the fermentation test, you're actually going to have 15 media tubes and then you're going to have your 4 bacteria. So make sure that you have the right amount of tubes. With the 15 tubes that we have, this one's extra, okay, you have 4 that are going to be used to put the bacteria in, and you're going to have 1 control for each type of sugar. So the reason that you have 15 is you're going to have dextrose, which is the same as glucose, you're going to have sucrose, and you're going to have lactose. Now glucose is a simple sugar, the other two are disaccharides. And so it's going to break, most bacteria will break down the glucose first and be able to ferment that. If you can't ferment glucose, most likely you can't ferment sucrose. But some will ferment a simple sugar but not ferment a specific disaccharide, or maybe it'll only ferment a different disaccharide. It might prefer the lactose over the sucrose, okay? And then you're going to have the controls for each sugar. So make sure you have yourself organized, because in this particular test, like I said, you have the 15 fermentation tubes and you have your 4 bacteria. Simultaneously, you might also be doing other labs such as the urea, the urease, or the litmus milk. So you might have a lot of tubes to deal with in one class period, so make sure you're very organized, and as a group, you split the work up evenly, okay? Maybe one student will do this bacteria and put it into these 3 tubes, okay? And then a different student does this bacteria and puts it into these 3 tubes. So share the work. Now, the first thing you want to do, obviously, is label your tubes. Make sure that you label them not with just the bacteria's name, but also your group name, group number, something to identify yourself. Because what usually happens is we condense everything in the incubator and things start to get mixed up, okay? So we have our 15 tubes, we have them labeled. Label the controls, C or something like that for control, because you're not putting bacteria in there. So then we're going to be going from here into these tubes. So as always, we're going to use our safety. Put on our gloves and goggles before we ever light our Bunsen burner. Light our Bunsen burner there. And so remember, I already have these labeled, and I know that I'm going from this bacteria, and I'm going to go, and I have the sucrose here, I have the dextrose here, and I have the lactose here, or whichever order you would have them in, okay? Just make sure that you know what you're putting into them and what sugar it is. So I'm going to take this bacteria and put it into these 3 tubes. I'm going to take the second bacteria and put it into these 3 tubes, and so on and so on. Well, I'm not going to do all of them, I'm just going to show you one. Okay, so as always, we're going to use our same aseptic technique of flaming our loop first. Okay, as that's cooling. Going to go ahead and take off our other 2 tubes, our lids. I'm going to flame those. Oop, didn't let it cool off long enough there. Okay. And then we go into the tube. What you want to do when you put it in there is not jiggle it too much, because you're going to introduce oxygen and maybe add a bubble. Okay, I got a big huge goober on there. Alright, so you're not going to do what I just did. I'm going to let my loop cool long enough. If you were to do that in real life, it's going to kill the bacteria and it's not going to grow. Okay? Ooh, that was nasty. Alright, so I'm going to put this one over here because it's done. Okay? You would repeat that procedure with all of the sugars, so putting this bacteria into the other 2 sugars, then this bacteria into the 3 sugars, and so on and so on. You will not put any bacteria into the controls, because those are just going to show what they originally looked like. Okay? You will still, however, put them in the incubator so that they can be a true control. So the other thing is, before you actually inoculate them, make sure you observe every single tube. Notice if there's an air bubble in the Durham tube already, because if there is, you're going to have to account from that at the beginning. So if you notice an air bubble, kind of estimate the size of it. So the next time, if there is a bubble, a gas bubble, not an air bubble, a gas bubble, and if it's bigger, then you can determine gas. But if it remains the exact same size, then it's not really gas production.